first of all, thank you for the opportunity to present today. And I'm really happy that so many people excited about this topic. So now on this workshop, I'm really excited myself. So I hope that it will be interesting, and I hope for questions afterwards, because just please remember, there is no stupid questions, only stupid answers. So you just please ask if something not clear or you want to know more. Of course, there are a lot of literature about this topic nowadays, and I try to make in this presentation some summary. Of course, I might be biased because I'm working in this field myself, but I know that the slides will be shared afterwards, so you can check the references and some links I also provide at the end. So, and yeah, so as I stated in the title, so I think that's a good definition, like how this artificial intelligence can improve, let's say, make our analysis faster, better, stronger. But first, because well, I'm not sure about knowledge of the audience, I just want to make first some intro to the artificial intelligence and the field. So of course, artificial intelligence by itself sounds really exciting and promising, and some way futuristic, but we should understand that, well, when you hear someone talks about artificial intelligence, it most probably it's about machine learning algorithms or about deep learning. So in deep learning, it's about neural networks, which consist of the neurons. And if you simplify it to the one unit, so basically one neuron is just, it can be just basically linear regression. So you have some input parameters, you have some output function, and in simple case, one neuron can be just represent a linear regression, or if you change it a little bit, it can represent just logistic regression. So, and that from one hand, this is quite simplistic, but from other hand, this allows you to build really complex models. So let's say if you want to connect many neurons and many layers together and input some data, you can see how they activate and how the data propagated through your neural network. And that gives you, based on the, yeah, you can make it bigger, you can make it wider, etc. So I will mention later a few examples. And this is example, it's like one-dimensional data input, but of course you can use two-dimensional data. This is just a nice example where you have a handwritten digits, so you have a 2D image, and then every pixel of this image is an input parameter to our network, and you can see how that propagates through these layers, and some neurons are activated. So basically it's a combination of many linear regressions or logistic regressions, and at the end it can allow you to analyze really complex data and perform analysis really well. And of course there are different types of architecture, as I said, so I will cover a few examples which I think are really promising and interesting in the field. And I hope that in the coming years will be more and more research based on these networks. But you can see that there's like actually quite a zoo of different architectures. Some of them just really simple with one layer, some have more layers, some have some convolutions, some really have end-to-end connection, or let's say the last example, Ernan, it has some memory even unit. I'm not going deep in details. That one is mostly used, let's say, in some language processing analysis. But coming to the topic of this talk, so why it's faster? So, and why deep learning? So, and it's quite simple explanation, because we all know that, especially with omics data, we are having every year and more data available, and we need better and better algorithms to analyze this. And you see that actually deep learning allows to analyze such data much faster compared to the classical algorithms. And why this is the case? It's kind of I would say, positive loop, because it started that deep learning can be simplified to these simple units like neural networks, neurons, and the simple operations of the matrix, basically multiplication or vector multiplication. And the GPU cards were really good for this. And once it became obvious that GPU card can speed up this process, then companies, in this example, NVIDIA, they started to develop better and better GPU cards for such type of the computation. And that actually even accelerated even more the performance of the deep learning, and then it forced companies to develop even better GPU cards and etc. So and then that's why deep learning can be really fast. And you can see it as examples for the image processing. So that there are so many algorithms in different type of medical image analysis, and there are a lot of companies which already registered their AI or deep learning based algorithms, because again, so that gives really high performance for medical image analysis. But if you think about the application for the fast omics analysis, so the first, I think that the most obvious example and which came in mind, that's about imputation of the data. And I made some summary of the algorithms where they can be used. It's about genotype data imputation, it's about a gene expression imputation, about epigenomic data, proteomics. I haven't mentioned it here. Again, so just remind that the slides will be shared afterwards. You can check these tools if you're interested in. So, and just I want to focus your attention that a lot of methods are based on the so-called autoencoders. So I will cover this later. And if you really want to go deeper in details with this review, I highly recommend this really nice paper. You see reference on the bottom. Yeah, so faster. So it means that GPU cards and the deep learning computation brings it to the next level, where we can just save the computational time on the scale from basically from years to hours and days, because on GPU computation can be really fast. And well, in principle, GPU cards can be really cheap. Well, some of them are really fancy and might be expensive, but nowadays to perform some simple experiments, it's not necessary to buy a really fancy GPU card. So what about better? So why that type of the algorithm becomes so popular and outperform state-of-the-art methods? Well, but that's, in my opinion, that's simply because there is a mathematical evidence, which is just called universal approximation theorem, which says that neural network with actually even a single layer is able to approximate any function in the universe, basically. Only one point, of course, that we don't know how many layers and the architecture of this neural network. That's why, as I said before, you have so many different types of the neural networks, each of them trying to find this best solution for your specific case. And therefore, if you think about this machine learning or deep learning applications, so basically, if you have any of these low-level problems like clustering, dimensionality reduction, classification, or regression analysis, here you can solve this problem based on this theorem in the best way with deep learning or neural networks. And as I mentioned that in the imputation part, that this autoencoder, that's one of the type of the neural network which I want to focus more your attention in this presentation. So this idea is quite simple. This is example again, so with the hand-written digits, where you have some really noisy data, and this network, what it does, it just first reduces the dimensionality towards like latent space. So we'll say from like hundreds of input parameters to just a few or tens of them in latent space, and then tries to reconstruct it in the decoder part to the original image. And by some manipulating of the training, during the training and some parameters, you can actually achieve that autoencoder learns just a global pattern, and that's why your output will be denoised in this way. And the same type of the architecture using you widely use now based on the examples I showed you for the imputation of the data, because once you train it, it can impute or denoise your data really fast, like tens of times of magnitude faster than classical statistical method. And this is just, this is example with actually denoising for genotype data. As you see that, for instance, in input, you have missing values, and as output, you have already imputed data. So, and the accuracy of such method is quite high. And the speed of this such type of algorithms also, as I mentioned, due to the GPU computation and such type of the optimizations is really fast as well. Another example is this is imputation for specific location. So this is a really nice paper. I think this was published recently. This is Biohive link. That's about the multi-etic actually imputation, because you know that the genotype information is might vary, and if you train the algorithm to impute the data, then you can overlap the specific region in this case to perform the trans-etic analysis. It's really also interesting application, and I suggest also the paper to read if you have a similar problem. Another example is actually with this gene expression. So again, so as I said, it can be used for denoising, but also here, that's a nice example that you can actually manipulate this latent space and force the algorithm to learn specific pattern. So, and as well, because I think that people working with this type of the data know that the data can be quite noisy. This type of the pipeline can be used to denoise this data, and it's again based on that idea of the autoencoder neural network. Another example, if you use a methylation data, so you can not only train for denoising, but you actually can use this latent space, which is much smaller than your input parameter, to explore your data, and also apply the method like t-SNE or UMAP to see that does your network capture a specific pattern. So in this example, with methylation data, the latent space represents, let's say, healthy clusters of healthy and people with cancer. And what is interesting that using the method for explanation of your prediction accuracy, I will mention this method sharp later in the presentation. So you can actually see which of the CPGs are important for this clustering. So you can go back to your raw data and see that some CPGs are more contribute to the healthy subject and some of them to the cancer. So that's also interesting application of the autoencoders. Another actually type of the neural networks, which I also found really interesting, it's a new type of the networks for survival analysis, where you can integrate the multi-omics data as well for basically Cox regression, but that type of the network, they can approximate even the interaction between parameters. And let's say if you're also working with imaging data, then you can combine together such networks without any pre-feature selections. And if I can recommend a few really good examples of the pipelines and papers, it's quite recent papers about spatial single cell analysis, so which covers really amazingly like the whole pipeline written in Python from raw data to some interactive visualization and statistics, and also availability to run deep learning methods. So this is one example, and another example is EMAP. That's a bit more busy and complex picture for the single cell dataset analysis, but basically what it does is also does denoising and also batch effect correction. So again, so just check the slides later. You can see the reference and check the paper in more details. But now coming back to the more topic which I myself interested the most, that's about the problem with the black box. That's probably most of you heard about artificial intelligence, that it's not explainable, and the problem is, especially in the medical application, healthcare, in the omics analysis, that you want to know why you achieve certain performance. And there are actually, it's not anymore really black box, because there are a lot of development around. And one example, as I said, is Sharp library, which basically brings the model from black box to explainable algorithms. It applies for machine learning and deep learning methods. There's a nice also paper with application for the electronic health records data, so where you can see which actually data point and parameter are the most relevant for your prediction. But of course, we want to apply it for omics data, and there was like a few years ago a nice review that how we can actually, based on our knowledge, already biological knowledge, how we can improve and make a new type of the neural networks. And that's GeneNet framework example, which I'm myself involved, and this paper you can also find on BioHive. And this is a really interesting idea, how you can integrate biological knowledge. So basically you start from genotype data, so that's data you need for this network, but the rest are built on the public available data. So you can integrate knowledge about how these SNPs are connected to genes. Then you have a gene layer where each node represents just gene. Then you can connect these genes to the pathways, and actually that's the whole library integrated in this pipeline. So from the GTEx, Allen Brain Institute, Keka pathway annotation, and actually that I was thinking when I was in the first presentation today, I was thinking that actually that network information was really interesting, that it's also possible to integrate in such network architecture. And at the end, so basically after you trained, you can reverse the network and see, for instance, which layer are the most important for your prediction. So basically you can get a gene-based Manhattan plot, or if you integrated information about that you connected your neurons, that they represent a specific cell types, you can see which cell types enriched and the most important for the prediction. And of course, that such ideas can be used not only for genotype data, for microbiome data. And as I said, our idea, of course, a bit futuristic, that to be able to build something like this, but of course, we're still not yet there. But for instance, example, which I wanted to mention, that we are working together with Bimura and actually Joyce, that in multi-omics analysis, where we're building similar neural networks, but where we integrate gene expression, methylation data, and also you can even integrate some covariates and correct your analysis, and then see how this covariates integrates with your interact with your methylation data and gene expression data. So to summarize about beta, so that the analysis can be more accurate, based that the neural network can basically approximate any function, then the neural networks can capture more complex pattern. And nowadays there are a lot of algorithms and development that to make them interpretable. And the last part I wanted to talk about is about making it stronger, because you can say that yes, so that all these algorithms are really nice, but we know that the effects are really small, and we want to detect them. And normally we do this huge consortium-based analysis with the meta-analysis, and we can't share raw data. And here comes another project which I'm also excited and involved in my own research, which calls the federated learning. So that's basically a classical scheme for the GWAS meta-analysis, when you have a central site and then you collect data from another cohorts based on the summary statistics. But with deep learning analysis, nowadays, it's possible to do the same using specific protocol and frameworks, where the network, what I showed you before, will be integrated in local cohort, but the data will not be shared. It's only will be shared some parameters over the network, and then and then the results will represent basically like a pooled analysis without sharing the raw data. And idea is quite simple. There are different ways to do this. So you have a central site. You can see this workflow that you can distribute your model to other sites. They run the local analysis, then they share to the central site, and then central site aggregate the model results, and you just keep repeating. So basically, you just need a good internet connection, which nowadays is not really the problem. Or you can even run it in the peer-to-peer model, so where there's no central site, everyone aggregates everything, and then at the end you just summarize the network. So that's more about, again, so some details of the implementation and about let's say, the computational burden for the internet connection. But at the end, you will get the result the same way as you have all data in one place. So basically pooled analysis without sharing raw data. And there are quite some development in the deep learning community. This is example of the open source framework to do this, mostly focused on the image analysis. And there's a special Python library to do this. And the companies involved in such analysis, let's say NVIDIA, they have a pipeline for the image analysis again. But we are collaborating nowadays with the NVIDIA to develop this pipeline for omics analysis, for omics distributed analysis. And to summarize, so that basically that application new method can accelerate our research, while it can make us stronger. So also developing a new method based on the expertise and data. So that I think the role of community to bring knowledge to the artificial intelligence or deep learning frameworks, and that can not only accelerate but also give more knowledge and insight in the analysis. And of course, distributed learning will change the way how we do collaboration and work within consortiums. Yeah, if you want to recommend the frameworks, of course, nowadays all this deep learning mostly done in the using Python language, and this is the most famous frameworks to do this. Again, you can check slides later. If you can just use some time for self-PR, just please, if you're interested, for instance, in the microbiome data. So we just published recently within COST ML for microbiome action review paper. And of course, also we are doing a lot of in another COST action consortium network, so Gemstone, and also within Charge Consortium. So if you're interested, you can always join and read our also results of research. And I also leave a few links, so you can check it later and you can play around with some deep learning and machine learning, just to give impression if you don't have any knowledge. So it can be a good start to understand the intuition behind these methods. And with this, I want to thank everyone. And please ask your questions. As I said in the beginning, there is no stupid questions, there are stupid answers. Thank you.
Thank you very much, Jenna, for your very nice talk and overview of all of the methods for AI. Please, if you have any question, just raise your hand or type it into the chat. So for me, I'm a biologist from background, and I'm always very impressed with all these computer tools. So these last links that you sort of gave, that is a good starting point, but is it always, when you want to do this kind of stuff, do you really need a lot of background for this AI analysis?
Well, I think nowadays it's really simple. So you need to know, I can't say that you don't need to know anything, but definitely you need to know to have some scripting examples and about program languages. So, but nowadays with the libraries I mentioned about, especially the one, the Keras, it's really so it's really easy to run a simple analysis. Of course, if that doesn't work and you need to go deep in details, that that can be an issue if you don't have experience enough with this. And another thing, if well, actually, the most of this analysis nowadays is like 80% is about data cleaning and QC and preparing your data for analysis. Because if you compare that the how many lines you need to write some neural network, it may be like sometimes it's 5 lines of the code or 10 lines of the code, but then it takes like 100 lines of the code to prepare your data for the input. So yeah, that's
quality of the data, of course, is very important. There are several questions. So to begin with the first, is this modeling affected by the degree of dynamics, such as intransigent proteomics and metabolomics?
Just to make it clear, it means that just to understand the question better, so it means that this data is really dynamic, and that's why if you combine together multi-omics, let's say like gene expression with more dynamic, or like genotype data with more dynamic data like proteomics, can introduce some bias. Yeah, I think that's, but that's the same as for any type of analysis. Let's say we wish to have a data, especially for dynamic like proteomics and metabolomics, like measured, I don't know, like every hour based on some device from your finger, I don't know, but that's not possible, of course. And I think that I actually think that such type of the method can capture the pattern if there is some global pattern. So let's say to remove the bias and noise, this noise of these dynamic structures, better than the classical statistical method.
Yeah, but I can imagine that also here it is important to choose your data right. So if you have your target tissue, then only use your target tissue and not all the other data. Maybe that's also important, because dynamics, of course, in ours, but also dynamic in different tissues, for example.
Yeah, but for instance, that the network or architecture with these explainable nodes, for instance, what is interesting about this type of the analysis, so if you do some pathway enrichment analysis, classical based on GWAS, then you have to correct by number of pathways you tested. So basically you can even overkill your results based on this. But in a neural network, if you add more neurons which represent some pathways, so if they are not relevant for the analysis, they will be just left out, basically. So in this way, the power can be higher, so you can actually pull as much information as you want, and then the idea that the network can sort out which one the most important.
Okay, so question: how can we use TCGA cancer data sets like DNA methylation, gene expression, and copy number variation data combined together in deep neural networks models?
Well, I think that relates to the project what I mentioned, that we are working actually on in the BBMRI and together with Joyce about this multi-omics integration. So basically, I think that maybe the good example about that in neural network, you can have easily different type of the channels as input channels. Let's say if you analyze the image 2D image, you also have RGB channel for different colors. So, and the same approach can be used in neural networks, that you can just use a different type of the data as a different channel, and then it's actually up to you to decide, do you want them to interact from the beginning, or you want to first, let's say, go deeper with your layers and only allow them interact on the, let's say, on the high level of the features extracted from these data sets. So I think, but of course, you need quite some data or outcome to do this. I think that if it's for the cancer, I think that's actually a good example where you can much easier to detect the difference between case and control than compare, let's say, to just a classical continuous trait of the human. Yeah, so I would say that it's just with a different channels approach.
Okay, so next question is, could these autoencoders be used to solve common problems like missing data imputation?
Yes, that's definitely, that's actually a few examples I showed that now used a lot. So, and yeah, that's I think that one of the ways to go is like if you have some problem about this. So I think that autoencoder with missing data, that's that would be the way to go.
Okay, and the last question I see in the chat is a very long one.
Yeah, but
Maybe I missed it, but what is the advantage to perform GWAS MA with deep learning networks as compared to combining summary statistics from a standard linear or logistic regression? So he's asking about statistical advantages. Would it be a better model fit? Yeah.
Yeah, I think that well, in the first approximation, let's say that if you have the deep learning networks, and well, that's a good question, and really maybe a long answer. I think that the advantages could be in a few things. Okay, first that well, deep learning by definition is better than by like a theoretical definition is better than linear regression, logistic regression. Of course, second that it can model interactions. So if you assume there is some epistasis for genotype data, and definitely for like metabolomics and proteomics and I know other omics, there is definitely interaction, and that network can model this better. And yeah, and I think that in a way in the future that will replace the PRS approaches for polygenetic risk scores, because that can be just more accurate, but that will require, let's say, just a larger data set to train the networks.
Okay, so, and I think the last question from Ingrid, are you familiar with the H2O AI platform? And if yes, how does it compare to the platforms you are using?
I'm not sure. I definitely heard about this platform, but I think that's some high-level framework. So basically, they trying to make it easier to build a network. So I think that if I'm not mistaken, so that's the main difference, is that it gives you easy to start, but then if you need to change something, it might need you can't do this basically. So I think that if you let's say to learn from scratch like Python, let's say, that's a bit higher input, like learning curve, but then you will be more flexible, and then with such frameworks.
Okay, well, thanks again, Jenna.